Today's topic is about the respiratory system development, gastrointestinal tract development, and nervous system development. There's quite a lot of slides, but I will show you the most common variations and some special cases. You already saw many slides during the demo class, so I will go through the slides that are a bit different, a bit unique in a way. First slide that we will check, lung development in pseudoglandular stage. So we will start with the respiratory system first because it's the shortest topic for this class. So lung development in pseudoglandular stage. I will share my screen with you also so we can see everything. So, as we can see in the slide, that's one way how to understand the general idea, what you might see also in the colloquium, but I will start with pictures again. So, first of all, there's different slide variations. Some slides are a bit brighter in coloration, so more pinkish coloration, more eosin is present. Others are a bit more darker, more basophilic. The darker slides belong to sheep. So sheep's lungs are a variation, and human slides are a bit brighter in color, but you do not need to differentiate the weeks for this slide. If you remember lung development pseudoglandular stage, it will be okay. So first things first, I will zoom in. So I will take this slide and zoom in at a random location, more or less here. So first thing that you can notice here are developing bronchi. So this is a developing bronchus. That's number one, developing bronchus. So developing bronchus. Developing bronchus. And about these bronchus, so developing bronchus is, as you can see, it's not really labeled in the structured list, but you need to think logically here. So bronchi are developing in the lungs. So if you see large walled structures that have also quite thick epithelium, maybe some muscle tissue also developing, these are developing bronchi. So developing bronchus, this is definitely mentioned also during the demo class. You also see a lot of small openings. So these small openings here with cuboidal or columnar epithelium, these are developing bronchioles. So number two, developing bronchioles. So developing bronchioles. So these smaller openings with cuboidal epithelium, developing bronchioles. What you need to be careful with. So the thing is, lungs in pseudoglandular stage do not have alveoles yet. So no alveoles. And the smallest, I would say, smallest pathways within the lungs would be the bronchioles. So these developing bronchioles until terminal bronchioles. Technically, they develop until terminal bronchioles, and only in later stages we will start to see respiratory bronchioles, alveoles in general, alveolar ducts, alveolar sacs. So that will appear much, much later. So pseudoglandular stage typically has the specific look. So bronchi and bronchioles are here, no alveoles, and a lot of mesenchyme. So this light tissue, number three, between the bronchi and between the developing bronchioles, mesenchyme. A lot of mesenchyme. Mesenchyme. So there's a lot of mesenchyme. A lot of mesenchyme between the bronchi and developing bronchioles. So mesenchyme. I don't know why in the structure list it's written there's glands. No glands are developing here. No glands. You won't see glands here yet. They will appear in much later stages. So no glands yet. But bronchioles are developing and bronchi are developing. That's the main idea. What else? Blood vessels. I will show you two types of blood vessels. Number 4, that's an artery. Number 5, that's a vein. So arteries, veins, capillaries, but blood vessels. So 4 is an artery, 5 is a vein. There's different types of blood vessels, and please notice the following. Blood vessels have endothelium, simple squamous epithelium. So inside has the simple squamous epithelium always. The amount of muscle tissue is quite variable, but the epithelium is the first thing that can help you to distinguish this. So simple squamous epithelium only in blood vessels. No bronchi and no bronchioles have any blood vessels yet. So remember this, be careful. Also, typically around blood vessels, you have a bit more mesenchyme. So that's another specific detail. There's a lot of mesenchyme around blood vessels, a bit more than around bronchi or bronchioles. So more connective tissue, more mesenchyme. That also can help you a little bit. So mesenchyme. And then there's another thing that can be labeled. I see in the slide description, it's not that well described. Please notice this. I will use a bit different color with orange. You can see there's a bronchiole that's splitting into two. So as the lungs develop, so bronchi and bronchioles are growing, they split into two. So this splitting in two has a specific name. So I will write number 6, dichotomic division. So number 6, dichotomic division. Dichotomic division. So that means splitting into two. So you can have a bronchus that splits into two bronchi. You can have bronchioles that split into two. So dichotomic division. If you check the specific bronchiole, it resembles a toy called fidget spinner a little bit. So it has a similar look to it. And of course, please do not write that there's fidget spinners in the developing lungs in the colloquium. That would be a mistake, definitely. But dichotomic division, this also can be asked in the colloquium. So splitting into two, dichotomic division. And that's the main information about lungs. So that's the main info. It's one of the simpler slides for today. The tricky bit is about bronchioles and bronchi. So remember, developing bronchus, developing bronchioles. Bronchi are quite big, large lumen, relatively thick walls. Bronchioles are small with simple cuboidal, simple columnar epithelium. Okay, I hope this part is clear of the slide. I will show you the microscope. So I'll switch to the microscope. I will use the green arrow in the microscope to show the structures. So in the microscope, it looks something like this. So this is a slide also showing lungs in pseudoglandular stage. If I zoom in some of the regions, let's take randomly, any of these regions. Let's zoom in here randomly. So you can see there's a lot of mesenchyme. So mesenchyme dominates here. So a lot of mesenchyme is here as I'm pointing at with this green arrow. So mesenchyme. And most of these small openings, you can see cuboidal, simple cuboidal epithelium developing bronchioles. So these openings are developing bronchioles, basically all of them developing bronchioles. In this slide, if I go through it, I don't really see any clear bronchi. So there's no bronchi in this slide, only bronchioles. Maybe this is a very small bronchus due to the thick epithelium, but sometimes in slides you won't see bronchus at all, so be careful. I'm showing this variation so you can be ready for the colloquium. Maybe this one, you see it's much bigger than all the other openings. Epithelium's a bit thickened. There's even muscle tissue appearing here, but this looks more like a developing bronchus. Typically, the lumen is wavy, so wavy-looking lumen. So lumen has this wavy texture or wavy contour, as I can say. What else? If we move through, of course, in the mesenchyme, you might spot some blood vessels. I need to zoom in a bit closer. You can spot some erythrocytes, but the main thing, endothelium. So there's a capillary here. There's a small blood vessel. You see endothelium here. So blood vessels can have erythrocytes, but check the endothelium first of all. Endothelium, but that's most likely a blood vessel. It looks like a small capillary. So capillaries, venules, veins, arteries also can appear. And what else? About this dichotomic division, if I move through the slide, yes, we can start to see dichotomic division. You can see the splitting into two. There's a bronchus that's splitting in two here, and there's bronchioles that split into two. So dichotomic division is present. And one more tricky thing, please remember, lungs from the outside are surrounded by what? So what surrounds the lungs from the outside? Who remembers this cover around the lungs? Pleura. pleura. This is pleura. So visceral pleura or just pleura development. Pleura. So sometimes you might see the pleura. I will add an asterisk here. So pleura, developing pleura. So pleura, sorry, pleura, spelling mistake. So developing pleura. So that also can appear. Not in all slides, but in some. So here you need to use your logic. So this is a relatively simple type of slide. Next slide, we will check development of the small intestine weeks 8-9. So development of small intestine weeks 8-9. So next slide, development of small intestine weeks 8-9. So mouse, development of small intestine weeks 8-9. So again, I will go through the pictures and then we will check the slide. So that's the pictures. Let's find the small intestine. Okay, here. So let's check this image and let's talk about the small intestine. First of all, structure is very similar to mature intestines already, but there's multiple differences you need to be careful with. So first, let's talk about the things we can recognize. First of all, you can see there's simple columnar epithelium. So small intestine development, weeks 8-9, that's the slide name. And the first structure, simple columnar epithelium. So simple columnar epithelium. What's interesting here? So in this epithelium, two types of cells. Most cells are enterocytes. So like in histology, there's enterocytes already here, enterocytes. And there's also light cells that produce mucus, goblet cells. Number 3, goblet cell. So goblet cells also appear here. Goblet cells. So goblet cell. Then what's unique here? You can see the connective tissue. Technically it's mesenchyme, but what's special here? So in the intestine during this 8-9 weeks of development, mucosa is still connected to the submucosa. So we use a term connective tissue of the mucosa and the submucosa. So connective tissue of the mucosa and the submucosa. That's number 4. We mentioned that this connective tissue is connected. Both layers, both tunics are interconnected. And why? What you should note, I will write in brackets, no lamina muscularis mucosae. No lamina muscularis mucosa. Here you don't have a smooth tissue layer that separates mucosa from the submucosa. That's a difference when compared to mature intestine. And the second detail, no crypts, no intestinal glands, no crypts yet. There are some slides where the intestine is a bit older. You might see some crypt fragments, but here in most slides, no crypts yet. So no intestinal glands, no crypts of Lieberkühn yet, no crypts. You might see openings like these ones. That is not a crypt, it's a fold. So it's a fold that comes from the intestinal villi. So folds appear here, but no crypts yet. Then also I mentioned briefly, you still have intestinal villi. So number 5, intestinal villus. So number 5, intestinal villus or developing villus, developing intestinal villus. So you see intestinal villi. You also see fragments inside the lumen. So these are still intestinal villi, just smaller fragments that are inside the lumen. So intestinal villus. What else? Now the tricky bit. We have tunica muscularis. Check very carefully where I'm pointing at. So this number 6 is tunica muscularis. So 6, tunica muscularis. And there's 2 layers already. Number 7, that's inner circular layer. And number 8, outer longitudinal layer. So there's two layers already. What you need to be careful with, inner circular layer is always the thickest layer, so it's much, much thicker, twice or three times as thick as the outer longitudinal one. This outer longitudinal layer can be only a single cell thick, so it's very, very thin. And sometimes students mistake this outer longitudinal layer with the serosa, but it's a separate layer inside the muscularis externa. What about the serosa? Yes, it's here, but it's super, super thin. So very, very thin layer. So number 9, tunica serosa, super, super thin. Tunica serosa. So this tunica serosa is very, very thin. You might just see this mesothelium, so this simple squamous epithelium on the surface, but it's a very, very thin layer on the outside. Very, very small, very not that distinct. In some slides, of course, it can be a bit thicker, but it depends on the location. Typically, it's very, very thin. And many slides, it is also damaged. You might not see the serosa at all. Then, what else? If you check the inside of the intestine, you might see some epithelial fragments that are broken apart. For example, here, there's also maybe some fragments like this one. I'll put number 10 next to these small fragments. So these are epithelial remnants after recanalization. So after recanalization, so epithelial remnants after recanalization. So epithelial remnants after recanalization. And what's interesting, so recanalization is a process where the lumen reappears again within the intestine. So what you need to know from theory, during embryonic development, the whole GI tract has a lot of proliferated epithelium. So there's so much epithelial cells that they fill the whole lumen. So the whole lumen is filled with epithelium for a short moment. And later, a new lumen appears. So these epithelial cells die off, and a new lumen reappears. This is called recanalization. Closer to the week 8, 9, 10, this lumen has completely reappeared, and the dead epithelial cells will form meconium. So meconium is a substance, I would say, the first fecal matter that comes from a newborn. There's also other substances that could be found inside, but meconium forms from these dead epithelial cells. And why do we need this recanalization in the first place? Why the intestine fills with epithelium fully? This is needed to differentiate structures like intestinal villi, the glands, the folds. So this process is needed so the intestine develops correctly. This is also found in other organs, like the nostrils. Nostrils also go to recanalization. I think external auditory meatus, some other organs as well. So this is quite a common process in other organ systems as well. And what can happen? If recanalization doesn't happen, atresia can occur. Atresia. So atresia. So it's not a structure that's from theory. So atresia means that there's an area in a whole of organ that doesn't have a lumen. So basically, you might have areas where the lumen is no longer continuous. As a result, the whole of organ is blocked off. It can happen in the intestines, in the esophagus, and other organs as well. And you can read about atresia on your own a little bit, but this is typically associated with the problems in recanalization. So these processes are connected. In general, that would be the main information. Remember, no lamina muscularis mucosa yet, no glands, so there is no cryptolibercune yet, but in general, structure very similar to typical intestine. Yep. Okay. I will also tell this to those who are Zoom. There's a question, what to do with children who have atresia in the intestine. So No, of course, if nothing is done, they will die. But typically, surgical procedures are made. So, for example, there's atresia in the esophagus. You cut out this atretic part where where the connection is lost between the two parts of the lumen, and you connect the esophagus back together. Or the same for intestines. You just reconnect the parts together. So it's like plumbing in a way, but pediatric surgeons do this quite a lot if this occurs. So typically surgical procedures are done straight after birth. So very, very quickly after birth, if it's diagnosed, then it can be treated surgically typically. Okay, that's about the small intestine. I will very briefly show you the microscope. I won't go through all the structures because it's very, very similar to the slides that are in studies, but some specific details. So, for example, here, small intestine development, you might see different sections. So in the microscope, as we go through, you might see longitudinal sections, also much larger sections of the intestine can appear here. Like here, for example, there's some other sections that are more cross-sectionally like, but if we zoom in, again, we see the intestinal villi with simple columnar epithelium. We can nicely see enterocytes. There's even some goblet cells that appear in the slide as well. Then these epithelial fragments after recanalization can also appear a little bit. So epithelial remnants or fragments after recanalization. Intestinal villi are visible. And remember, when we talk about the connective tissue here, so this is connective tissue of mucosa and the submucosa. So you name mucosa, submucosa together here. Then tunica muscularis, very, very thin. Inner circular layer is 3 cells thick, outer longitudinal layer 1 cell thick. Very, very thin layer, as you can see. Both of them form a lamina, they form tunica muscularis or muscularis externa, that would be the correct name. And then the serosa. This slide is interesting because the serosa is thickened here. You can see a lot more mesenchyme, some mesothelium. And one thing that you can also add as an additional label, it's not in all slides, you can see this area. There's blood vessels with mesenchyme in the wall of the intestine. This is called mesenterium. So developing mesenterium. So developing mesenterium, that's specific for some slides. It's not the typical structure that says, but when you have a question, what is this connective tissue connected to the intestine? That's the mesenterium. So developing mesenterium, you can see. In general, that would be it. So also relatively simple slide. Here your histology knowledge will be quite helpful in understanding the structures. So, is this slide okay? Understandable? Yes, there's a question. Goblet cells. So about goblet cells, you can only see them in high magnification. These are cells, yes, that stain lighter than the other cells. Like here you can see goblet cells in the epithelium. There's a goblet cell. Here there are goblet cells. So light cells that don't stain that well. So large light cells, like these ones, goblet cells. So those are goblet cells. So that's how you can recognize them. Same as in histology. So same as in histology. I see there's a question. Where is atresia? You do not need to label atresia. It's for the theory part of the colloquium. So you need to read what atresia is. It's not the structure in the slide. I mentioned it before. It's not the structure. It's a term that we use if there's a blockage within a hollow organ because recanalization hasn't occurred properly. Bless you. Okay, let's move on. Next slide. So this was the small intestine. We will check the next slide called liver development. So liver development. Liver development. I will try to find a good liver slide. This maybe. Oh, sorry. Es to pārzvanīšu pēc brīža vēlāk, ja? Es nevarēšu uz diviem. Uz diviem es varēšu, ja? Es divos varēšu. Jā, man ir nodarbība, tagad es nevaru. Davai. Sure. Sorry? Yes. Let's check here briefly. No, there's no liver. There are good liver slides. Let's use this one. So I will go first through the pictures because the liver can be quite tricky. But what about the liver slides? So liver development slides. So I will write liver development are variable. So some are taken from rats. So I will find the liver development. Some are taken from rats, some are from human embryos. Let's start with the human embryo slides. So, first of all, you might get some fragments of the liver, but to determine what is inside the liver, you need very high magnification. Let's start with these bigger slides first, and then we will check slides where we can zoom in into structures. First of all, here, this is liver. So number one, developing liver. So liver development. So as simple as that, we see the liver. We cannot distinguish the cells, so because we need very high magnification. Who knows in which cavity the liver sits in? Abdominal cavity, good. Abdominal cavity. So abdominal cavity. And who knows what lines the abdominal cavity? So I'm writing in the slide, don't worry. So I'm writing, so liver development. So liver is number one, two, abdominal cavity. I'm writing this also in chat. What covers the liver here on the outside? Who remembers from anatomy? Peritoneum. Peritoneum. So visceral peritoneum, if to be exact, but peritoneum. Here, if you know anatomy, that's very helpful. So peritoneum. Okay, what is this cavity? Number four. Thoracic cavity, good. Thoracic cavity. Number four, thoracic cavity. And what separates the thoracic cavity from the abdominal cavity? Diaphragm, good. Diaphragm. You can write always developing. For every structure, you can always write developing. So developing diaphragm. So diaphragms here. So because it's the thoracic cavity, what do you think? What are these structures surrounding the thoracic cavity? Rib development. Yes, developing ribs. As simple as that. And who knows what's in between ribs? What is there? So, any ideas? Skeletal muscle tissue development or intercostal muscle development. So development, skeletal muscle tissue. So yes, as simple as that. If you remember anatomy, it's very helpful in embryology. It's very helpful. This last part, it's a bit more tricky, but basically this is the developing shoulder region. So developing shoulder region. I will put an asterisk because it's typically not asked in the colloquium. It's not asked. But if you have any questions, what's there on the top? The shoulder region is developing. Which bones a bit unclear? Maybe the scapula, acromion, some other areas on this region develop here, but it's quite hard to distinguish. So that's why I'm putting an asterisk. That's about this slide. Now I will show you another slide variation. So let's check. I will find the rat slide, this one. So this slide, it contains the liver. So the liver is here. So the liver is here. So again, I will put number one, liver development. So liver development. And what is tricky here? So rats are a bit different than humans, but still the topographic anatomy is the same by idea, because there's both thoracic, abdominal cavities, and similar organs are located in similar positions. So what is important here? Again, what is the cavity where the liver sits? Abdominal cavity. Good. Abdominal cavity. That's number two. And what is this cavity? Thoracic cavity. Good. Thoracic cavity. Good. Then what separates these cavities? diaphragm, so developing diaphragm, that's number 4. Yeah, if I'm going too quick, you can inform me. Right. Oh, yeah. Then, what else is here? Any ideas what are these organs? Lungs in pseudoglandular stage. So lungs in pseudoglandular stage. Lungs in pseudoglandular stage. What is here, number 6? What exactly is there? Thoracic vertebral body. So developing vertebral body, developing vertebral column. Okay, number 6, developing vertebral body or corpus vertebrae development. What's behind the vertebral body? Here. Spinal cord, yes, developing spinal cord. As simple as that. So here, if you know topographical anatomy, where each organ is approximately located, it will be very helpful. Then, what's in front of the vertebral column? You see a blood vessel here. Any ideas what this could be? It's an artery. Which artery? Dorsal aorta, good. Dorsal aorta. So if you see artery in front of the vertebral column, most likely it's the aorta. And then you see a structure that has a weird shaped lumen. It looks like a tiny human. So if you zoom in, it looks like a very weird shaped lumen, thick epithelium. What that could be? Number 9. What is this? No. 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 In thoracic cavity, there's a structure that belongs to GI tract. What is it? Esophagus, correct. So developing esophagus. So number 9, developing esophagus. Esophagus can be seen in some of these slides, especially if you see something near lungs that doesn't really look like a bronchus. So developing esophagus. So esophagus can appear. Any idea what are these structures? Ribs, developing ribs. So you see developing ribs here. These fragments, these are developing ribs. Number 10, developing ribs. In general, we can continue with this on and on. There's skeletal muscle tissue, there's skin development, epidermis, dermis, hair follicles, that continues on and on. So if you get these rat slides, there's quite a lot of details you can go. And then what about the slide name? Technically, you can say, okay, there's liver development, but you can also name the slide a bit differently. This is a cross-section or transversal section of a rat embryo through the liver and lungs, for example. You can write in different ways. Basically, it's a cross-section of rat embryo, and you can write which organs you see: liver, lungs, spinal cord, the ribs, etc., etc. So this is another name that you can use for these slides if you're unsure. If you're unsure about the slide name, write everything down that you can see there. So, what exactly? In this case, these are only rat embryos are cut like this. No human embryo looks like that in the microscope. No human embryo looks like that. Most are rat embryos. What I'm going to show you. So I will show you the slide and I will zoom inside the liver, because in the slide description, the structures that are asked are only seen in the highest magnification. So for this, we will now check the slide under the microscope. I will show you which cells actually sit inside the liver. So I hope this is clear. And then let's zoom in. So let's turn the screen here. This is rat embryo cross-section showing the liver. And what's interesting here? So this slide is very big. This is the lowest magnification. And the problem is, as you move through the slide, you will not be able to see everything in detail. So there's a very large liver. you might see some additional tissue. Here, this slide is also taken from the human embryos, but I will zoom in closely inside this slide. I will show you maybe another variation that's a bit better. Where are the liver sections? Oh, this is a better section. I will show you this one. It's a bit better. So rat embryo cross section. So this is a very large liver. So liver takes a lot of space in the slide. This is just the liver, very large slide. We see the diaphragm and the lung development and pseudoglandular stage afterwards. And if we continue to move, we see the esophagus. So you see this unique shaped lumen, that's the esophagus. If we move further, you see the vertebral column, so this corpus vertebrae is here, aorta is visible. You also see the spinal cord. We will see the slide again because we have a separate spinal cord slide. So some of these structures can reappear in the same slide. Then, of course, muscle tissue, skin development, as we talked last week. But now let's zoom in into the liver. So if we see the liver, we need to use the highest magnification to see the cells. So I will zoom in. And let's check the cells. So first of all, what stands out here? But on the highest magnification, you will see a lot of cells. The first cell I'm pointing at, this is a hepatocyte. So developing hepatocyte. Where the green arrow is pointed, that's developing hepatocyte. I will also use the labels here. So hepatocyte, I will encircle here. That's developing hepatocyte. Number one. Developing hepatocyte. Developing hepatocyte. So, developing hepatocyte. Those who are attending, please write your names on this paper. And if you're coming in attendance on site, take the paper, write your name. On this paper, here. Okay. Developing hepatocyte. These cells look almost like adult hepatocytes. So large, not big cells, but relatively medium-sized cells with a nucleus, nucleolus. Some inclusions are present. So hepatocytes. Then, another thing. You will see that inside the liver, there's small groups of dark staining cells. So dark stained cells. These are hematopoietic islands. So these groups of cells, we call them hematopoietic islands. Hematopoietic islands. Hematopoietic islands. Or these also called nucleated erythroblasts. Hematopoietic islands, so nucleated erythroblasts. So these are blood stem cells. So these cells in the liver help to create new blood cells. Some will form erythrocytes, other cells will form immune cells, but they form new blood cells. And the liver has a lot of these groups. If you see here, there's a lot of these hematopoietic islands throughout the liver. So liver is one of the main locations where hematopoiesis happens. So it happens very actively during the second trimester. And when closer to the end of the third trimester, new blood cells start to develop more within the bone marrow cavity. So red bone marrow appears much later. But first, in the liver, a lot of new blood cells develop, specifically here. And because new blood cells are developing, you will see another cell. Number 3, megakaryocyte. So number 3, megakaryocyte. These are large cells that have lobe nuclei. Cytoplasm can be pinkish, can be basophilic, depending on the slide, but these produce platelets. So megakaryocytes also tend to appear inside liver tissue. So 3 things that you can label. You have the hepatocyte, hematopoietic islands that have nucleated erythroblasts, and megakaryocytes. These are the 3 cells that can be asked in the colloquium. I've seen megakaryocytes are not labeled, but basically they also are one of the blood stem cells. They produce platelets. These are very large cells with multi-lobe nucleus, resemble osteoclast a little bit, but that's a megakaryocyte. 3 cell types. Is this clear? You don't need to distinguish any blood vessels. There's no classic liver lobules yet, nothing like that. Bless you. So that's the basic idea here when we talk about the liver. Is the idea clear? Megakaryocyte, that's number 3. This big cell where I'm showing with pink color, that's megakaryocyte. This also is a megakaryocyte. This also. Yes? Number 1 and number 2, how to differentiate it? So number 1 is a hepatocyte. All the cells that I'm showing you with orange color, all of these are hepatocytes. First of all, they have a nucleus with a nucleolus. And you see these cells are bigger than the hematopoietic islands. Hematopoietic islands look like dark blue dots without any nucleolus inside. So hepatocytes are medium-sized cells. I'm using orange color to show you all the hepatocytes, which I will be drawing here for the whole day to circle all of them. All of these are hepatocytes, all of them. The small dark blue cells, those are nucleated erythroblasts that form the hematopoietic islands. Is this better or clearer? Yeah, okay. This is the structure that I'm showing. The cells are asked only in high magnification. So you can only ask liver cells very high magnification, only in very high magnification. I will zoom, yes? There's blood vessels and a lot of other stuff here. Those are not structures that could be asked here. There's some already developed blood cells, big sinusoids are forming, but you don't need to label blood vessels. Only these 3 cell groups, cell types. So hepatocytes, that's number 1. Hematopoietic islands, that's number 2. And number 3, megakaryocytes. I hope you got this, yeah? I will zoom out. So in these slides, we will always start on the lowest magnification. That would be this magnification. I will show you throughout the whole slide. If there's additional organs and structures, I will show you them. But if I'm interested in the liver, I will zoom in onto the highest magnification to show you the different types of cells. So liver is one of the rare slides where we need to zoom in to see the cells. But that's the general idea here. Next, I will show you some slide pictures, also depicting the liver. There's some slide variations that are quite tricky. For example, I will find this one. There's very rare slide variations. No, in the colloquium, typically, I don't like to use these slides, but this is a liver slide also. So here you have the liver. So number 1, that's the liver. This is the liver. And we also see some ribs. I will put letter R. These are the ribs, most likely due to the section. There's connective tissue, mesenchyme, technically skin also here, but you cannot distinguish any cells at all. So this is a slide of not that great of a quality. These kinds of slides typically won't appear in the colloquium. So that's one variation that also appears in e-studies. This is not for the colloquium. So it's just a variation of the liver slide. We cannot distinguish the cells at all due to the staining of the slide. And then what else? We will check the next slide. Next slide will be called, the physiological hernia or physiological herniation of the midgut. So next slide is this physiological herniation of the midgut. So in slides, it's approximately in week 10, because many of slides are taken from human embryos. But if you forget the week, it's not the worst thing for the slide. So the main thing that you remember, the type of slide. So physiological herniation of the midgut. I will put the slide also on the microscope. So this slide is typically shown on the lowest magnification. And I will briefly put this on the microscope and then I will switch between the microscope and the pictures. So, yes? You will get full points if the slide name says, yeah. Yeah, if you don't remember the week, in this slide, that's not a big issue. Okay, so I will put side by side two images. One is from the microscope, one is from these studies, the picture. So, as you can understand, we are looking approximately at this region. This is the lowest magnification. So this slide again is very, very big and that can cause problems. But let's start with the simple things first. First of all, with number 1, you can also see here, that's the umbilical cord. Slide name I already wrote, physiological herniation of the midgut. Please, colleague, read the chat. So, physiological herniation of the midgut, that's the slide name. So 1, that is the umbilical cord. Oh, sorry. Cord, umbilical cord. It has the same structures as in the second class. Wharton's jelly is there, blood vessels are the same, so it's typical umbilical cord. Then, what else? Here we see structures that I will label with number 2. So you see a lot of them also inside the human embryo, and here in the slide you see this quite a lot. You can write in different ways. You can write developing small intestine. That's one way. Intestine. Or you can also write, I'm checking, so developing midgut. That's also an option. So developing midgut. But developing small intestine, it's an okay term. What's interesting, technically we can ask the same structures that were present in small intestine slide. Epithelium, the connective tissue, mucosa, submucosa, muscle tissue around, but developing small intestine loops, that's the general name for this whole structure. So you can see that these small intestines are pressed into the umbilical cord, and this is the physiological hernia. So this whole area where the intestines are pushed in, that's the physiological hernia or physiological herniation. So all these intestines plus the umbilical cord, that's the physiological herniation. Herniation of the umbilical cord. Physiological herniation. I will write like this, yeah. Physiological herniation. Why it's physiological? Because it's reversible. And this happens because liver grows very quickly. There's not enough space for other organs, and they get mechanically pressed into the soft tissue of the umbilical cord. Later, as the embryo grows and the abdominal cavity gets bigger, the intestines are pulled back inside. And then the process is reversible in a way. Herniation also can be a pathology. After birth, if herniation happens, and that's quite a severe pathology because it can damage the intestines, also cause ruptures and quite a lot of other problems. But here, it's a normal process during development. That's why physiological herniation. What also is typical? You will see the liver. I will put number 4 here. Always there's a very big liver, so liver development. That's number 4. Liver development. So liver is visible in the slides. Liver development. And again, if you would zoom in into this liver, the same blood cells can be asked. Megakaryocytes, hematopoietic islands, hepatocytes. They look exactly the same as in rat embryos. They look exactly the same. That's why I won't repeat them. So very, very similar look to this slide as well. So this is the idea about this region. But what we else need to talk about? There's some other structures you can label here. So I will delete these drawings. We will move to different locations. Let's move here. So I will label here on the picture. And I will use the microscope to move to this area. So we follow here. Let's move to this area here. So If we move to this area, you can notice a couple of things. First of all, there's an organ you can see here. So glandular tissue inside mesenchyme. So here that would be, so glandular tissue in the mesenchyme, that would be here. We see developing pancreas. Developing pancreas. So that's the developing pancreas. It's very a unique location. You see developing pancreas. It looks like glandular tissue growing inside mesenchyme, and it's in the abdominal cavity. We will see pancreas in separate slides also, but this is how it looks in humans. So developing pancreas appears in this slide. Then let's move out a bit more. I'll use this delete function. Let's move a bit further. I will label 2 organs here, 6, 7, and I will move through the microscope also a little bit. So, I heard some comments already. So number 6, that's adrenal gland. Number 7, metanephros. So number 6, developing adrenal gland. And number 7, metanephros. As simple as that, we can see these structures. If we would move even further, we would also see some other structures. Control Z. We might see some of the others, additional structures. For example, we already know if we go even further, we would see the vertebral column. So developing vertebral column is also nicely seen. So if I use the green arrow, you can see the vertebral column development. There's spinal ganglia, there can be ossification centers. So we already have seen a lot of vertebral column slides previously. What's special here, you might have the spinal cord. So spinal cord development also. Spinal cord development. Spinal cord development. If we move to the side here, you will see developing spinal cord also. So spinal cord development. What else is here? You see a big organ inside the thoracic cavity made from muscle tissue. I will move here in the slide also. So if you see in the thoracic cavity large muscular organ, that's developing heart. So heart development. So 8 is developing vertebral column, then heart development. So heart development. In front of the heart, you see a very dark staining organ. So again, if I move in the microscope, it will look like this. This is the thymus. So thymus development, developing thymus. It also appears very nicely in many slides. So developing thymus. So developing thymus. So thymus development is here. And then, if we move in behind the heart, you see mesenchyme tissue. Here you see bronchioles and bronchi developing, lung development in pseudoglandular stage. Lung development in pseudoglandular stage. So, lung development in pseudoglandular stage. So again, here use topographic anatomy skills. If you understand the general location where each organ has to be located, in which cavities or which body parts, then this slide is not that bad. The tricky bit is, of course, this herniation. So this physiological herniation that you see here, that's a unique part that's visible during embryonic development. So be tricky with this location. Yes? It always happens, yes. That's a normal physiological process. Normally, when intestines develop, herniation happens because the liver pushes the intestines out from the abdominal cavity. And later, as the body of the embryo grows larger, there's enough space for the intestines, they get pulled in back. Okay, I hope you have the idea here. if this is clear, next slide, developing pancreas. I will put a picture, developing pancreas. I will show you to this slide first. And I will show you also the human slide. So developing pancreas. So we will see the slide also next week, because not next week. Next week there's a holiday. The week after that, yeah. We will see these slides again. I will check if I have a good human slide of the pancreas. Pancreas 25A. Just wait a moment. There are sometimes some limitations to the slide. No, okay, I will show you the rat slide then. So, okay. This is the rat sagittal section. This is a massive slide, but I will point at the area where the pancreas is visible here. So, in the first glance, it's very tricky, but it's actually not that bad. So, in the picture, I will show you the area where the pancreas is here. All of this is the pancreas. So in red color, that is the pancreas. So number one, pancreas development. And in the slide, it's here. So one, developing pancreas. So slide name is pancreas development. And one, developing pancreas. And to be exact, exocrine part. So exocrine part only develops here. No islets of Langerhans. Inside there's serous acini. So you see rings of cells, there's serous acini that are developing here. So I will put in brackets, developing serous acini of the pancreas. Serous acini develop here. There is mesenchyme also in between. So if you see connective tissue in between, I will put number 2 inside. That's mesenchyme. So mesenchyme, connective tissue that develops around or in between the acini. What else is here? I will use a bit different colors. So here with blue color, this is the liver. So see, liver is very large. So the liver is visible here with number 4. So, number 3, I mean. So number 3, the liver development. So the liver is here. And what else? I will use orange color. Again, you see developing small intestines. So here with orange color, so these are developing small intestines. That's number 4. So number 4. Small intestine. Sorry for the typo. And the slime is the same thing. With number 4, you see intestines typically, and the liver typically is much, much bigger. So the liver tissue is here. There's some artifacts also, but the liver appears as a separate, larger organ. And so pancreas in the rat embryo, it's found between the liver and the intestines. In humans, I showed you how the pancreas could look like, glandular tissue growing in mesenchyme. Pancreas can appear in other slides, like this physiological herniation of the midgut. There's also separate slides, and I will show you a separate picture. We still have a lot of slides to check, but that's the idea here. You need to focus on these areas. About other organs, we will talk about during next class. So there's quite a lot of organs we need to check separately.
Sorry, I have a question.
If you have a question, better write in the chat so it's easier for me to read. The audio here is not that great. I won't hear you that well. Better write in the chat if you have questions. Yes? No, you don't need to recognize that. Technically, you can try, but it's the rat embryo, so it's it's up for discussion. Is it really that specific lobe or fragment of the lobe? Orange color structure, small intestine development. So number 4, small intestine development. intestine development. That's the orange structure, small intestine. Okay. I will show you some pictures because on the slide place we don't have all the variations. I will show you how pancreas can look in humans. We have half an hour left. So again, I will use the pictures. I will delete these drawings and sorry. So mesenchyme is the connective tissue between the glands. So there's some pancreas slides where they're zoomed in. I will find it, this one. So this slide also shows the pancreas. So you have pancreas development, same numbers that I used before. And mesenchyme is the connective tissue between the pancreas tissue. So between the serous acini, you see these acini very well. Serous acini are present. But mesenchyme is the slight connective tissue in between. Here in this slide, you also see a liver. So liver tissue appears. No intestines in this fragment, but liver also. And if you zoom in this liver, again, all three liver cell types can be asked here. If you see them, of course. Hepatocytes, megakaryocytes, not in this slide maybe, but hematopoietic islands stand out. But that's the idea here. That's in the rats. So this is rat embryo. This is still pancreas development. Serous acini. So developing serous acinus. Developing serous acinus. Developing serous acinus. Okay, I hope you got this. What else we need to check? There's human slides. So if you want to find the pancreas in human slides, here it is, bless you. Developing pancreas is here. This tissue shows developing pancreas. So you see glandular tissue growing inside mesenchyme. That is developing pancreas. What is interesting here, there's also the stomach. So developing stomach and small intestines. So small intestine development as well. SE, small intestine. So one is pancreas development and stomach is developing, ST, stomach development. So stomach is developing here and small intestines are developing next to it. There's also lungs, letter L, lungs, vertebral column, and the trachea as well. This is a unique slide. One, here, number one, pancreas development. Yes, I can slow down, but unfortunately we have a lot of slides. I don't want to keep you half an hour after the class, so. Yes. So yeah, if something's missing, please ask. Yeah. Yes? Trachea. Let's see, small intestine. Yeah, just wait a moment. I will write down everything. Lung development. So these structures unique for this specific slide. Yes? Yes. Technically, you will see there's a plate of hyaline cartilage here in front. So there's a plate of hyaline cartilage. You will see this, like the trachea. Yeah, like in histology, the same idea is present. Anatomy. If you would zoom in, you would see these rings. But typically, this is a very, very rare slide variation. Okay, colleagues, if something's missing, please ask also in the chat. Yeah, thank you for mentioning. If I'm too quick, also, yeah, tell me so we can follow through everything. Yeah. Again, if something's missing, maybe some slides are a bit too tricky, we can check them also during consultation times before the colloquium. So do your better safe than sorry if needed, because there's a lot of complicated slide variations. We can go through the main ones, And quite a lot of these you will need to check on your own also. But that's why there's demo classes. There's a lot of slide information given here, and we go through the main parts also. Okay, so is this slide clear? Also, those who are online, those who are here. Of course, there's ribs also. As you can see, a lot of ribs are here, so you need to use your anatomy knowledge as well. So we can finish the GI tract finally, and we can start to talk about nervous system. So next slide will be neural tube development or development of neural tube, weeks 6, 7. Next slide will be this one. I will start with pictures first because they are simple. This is a relatively simple slide. Basically, it will be very similar to the spinal cord. Main difference, it's in longitudinal section. So longitudinal sections are present. So development of neural tube, weeks 6, 7. First things first, you see an opening. Canalis centralis. Central canal. Number 1, canalis centralis. So number 1, it's canalis centralis, you can see. And as you know, so canalis centralis is lined with some cells. During embryonic development, we call this darkened zone of cells the ventricular zone. So number 2, ventricular zone. Ventricular zone. From this zone, it will form ependymal cells. So forms ependymal cells. Ventricular zone will form ependymal cells. Then number 3. This is another zone. You see a lot of cells, but not as many cells as in the ventricular zone. We call this the mantle zone. So mantle zone. And it will form gray matter. So mantle zone forms gray matter later. So it will form the gray matter of the spinal cord later. But mantle zone, a lot more cells inside. And then zone here, number 4, this is called the marginal zone. So number 4, marginal zone on the outside will form white matter. Will form white matter. So canal centralis in the middle. We have ventricular zone number 2, mantle zone number 3, and marginal zone number 4. So these are the zones that are visible here. There's another detail. There's sometimes small fragments of tissue. I will basically create a drawing here on top. If you see thin layer of connective tissue on top, in picture it's very hard to sometimes see. This on top, it's also on the spinal cord slides. I will put an asterisk, pia mater. Connective tissue on the outside that would surround the neural tube and later the spinal cord, pia mater. That's number 5. I will put an asterisk. It's not in the slide picture, but in slides it can appear. So pia mater. So pia mater can appear in slides. Pia mater. Pia mater. Here we don't see skin. There's some slides where there's some additional tissue, but we don't see epidermis. We don't see dermis here in these slides. We can only see them in larger slides if we have the whole embryo. But you need to remember these 3 zones. That's the main detail here. Is this slide clear? There's also a similar slide variation that looks like this. It's very similar, the same idea. All 3 zones are here. If I go here, again, we'll go through the same numbers. 1 ventricular zone, 1 is canal centralis, 2 is the ventricular zone, 3 mantle zone, and marginal zone is outside, number 4. Same idea. So 1 canal centralis, 2 ventricular zone, approximately here mantle zone, and the marginal zone is very thin. That's number 4. Bless you. And here actually there's a very thin layer of pia mater, very, very thin layer outside. Here you might see small fragments of pia mater on the outside. So pia mater is in this slide. I hope this slide is clear. Next, we will check the vertebral column. So vertebral column will be similar, only difference, vertebral column development is in cross-section. Cross-section. So next slide, development of vertebral column. Development of vertebral column. What is interesting about this slide, here you don't need to remember the weeks because it's mostly taken from rat embryos. Also, there might be human slides, but most of the cases, these are rat embryo slides. If we zoom in, again, let's go through the structures. 1, canals centrales. So again, there's the central canal, canals centrales in the middle. The same orientation as in the previous slide. 2, you see dark stained cells, number 2, ventricular zone. So number 2, that's the ventricular zone. Dark part, yes, dark part, that's the ventricular zone. Again, same idea, dark staining cells. That's the ventricular zone. So dark staining cells around the central canal, ventricular zone. Then you see this butterfly-shaped area. This is number 3. We call it the mantle zone. So number 3, mantle zone. So it looks like a butterfly. So this butterfly shape, that's the mantle zone. It will form the gray matter later. So mantle zone. Then here, the zone that you also should remember, marginal zone. So number 4, marginal zone. So it will form white matter. If you remember anatomy, how the spinal cord looks like, then it's not that bad. Canal centrales in the middle, dark cells around ventricular zone, butterfly-shaped mantle zone, and marginal zones on the outside, light zone outside. What else is here? You see thin plate of connective tissue, pia mater. So number 5, pia mater. So pia mater appears in these slides. Pia mater. Pia mater. What else is here? You might spot dark groups of cells sitting next to the spinal cord. Also, this belongs here. This is also number 6. Developing spinal ganglions. Developing spinal ganglions. Developing spinal ganglions. That also appears in this slide. We will see separate spinal ganglion slide also, but this is here as well. Any ideas what is number 7? You should recognize this. What is this? Developing vertebral body. Correct. So developing vertebral body. Good. So developing vertebral body. Any idea what is number 8? Again, please. Process transversus, but there's a joint in between here. Rib development. Number 8, rib development. If there's a joint, it's most likely a rib, but I would count in also process transversus development. But this is a rib. You can actually see ribs. And then if you remember the vertebral column, You might see cartilage here on the outside behind the spinal cord. What is this? Number 9. Any ideas? Vertebral arch or arcus vertebrae? Developing arcus vertebrae. If you write developing vertebral column, technically it's correct. That's also correct. But arcus vertebrae, this is the arch of the vertebral column, located on the opposite side, on the other side along the spinal cord. Any idea what's here? Yes, developing skeletal muscle tissue. So number 10, developing skeletal muscle tissue. So developing skeletal muscle tissue. Which muscles are these? The back muscles, I would say. So these different kinds of muscles. Most likely, if they're close to vertebral column, these might be these not intervertebral muscles, but muscles that help to connect the different ligaments, different parts of vertebral column together. So the back muscles, to say it for short. But if you write skeletal muscle tissue, it's okay. And then a tricky structure that's not in many slides, I will put an asterisk because it's not always visible. This is truncus sympathicus. Truncus sympathicus development. This is an exception. It's not typical in slides, but if you see something that looks like a ganglion in front of vertebral column, that's truncus sympathicus. Truncus sympathicus. But it's a very special case. It very, very rarely appears in the microscope. Okay, I hope you got this image. Those who are also on Zoom, I hope you got this slide. If there's some structures that are missing or maybe unclear, please ask. We don't have much time left. I will briefly show you the microscope. So I will clear my drawings here. Then let's go to the microscope. So vertebral column is asked typically in slides where you might see other structures as well. Well, for example, let's take the rat embryo. Rat embryo cross-section. And if you check here, I won't go through all the structures here on the slide. We already checked the picture. Of course, we see the lungs, esophagus, but let's check here. The section is very, very similar. If you zoom in, again, if you say the spinal cord, canalis centralis is in the middle, around ventricular zone. So this is the ventricular zone around. Then a lot of cells sit in the mantle zone. So mantle zone is rich in cells, as you can see. And the light zone on the outside, this is the marginal zone. So 3 zones: ventricular zone, mantle zone, marginal zone. Same idea. Around pia mater, so the thin connective tissue on the outside, pia mater. Then on the sides, spinal ganglions. You might spot spinal ganglions separately. So there's some spinal ganglions. Vertebral column, as you can see, muscle tissue and other structures as well. So this is quite a big slide. It's very similar to other cross-section slides of the rat, but we might focus more on the vertebral column. We might focus on other areas as well, mainly on the spinal cord, as we can see. So this is just a slide variation. We have 2 more slides to check, and both of them are a bit tricky. Let's check spinal ganglion development. So developing spinal ganglion. That would be, I will find this slide. Developing spinal ganglion, and I will zoom here at the bottom. So developing spinal ganglion. So it can be asked in the rat slides, in the human slides as well, but developing spinal ganglion slides are quite interesting. I will briefly show them to you. So developing spinal ganglion here. There's some slides where we separately have these developing spinal ganglions. So I will use this one. And we will use the pictures. So yes, pia mater is the connective tissue that surrounds the marginal zone. Yes, that's the question you asked. So yeah, that's pia mater surrounds the marginal zone from the outside. Okay, spinal ganglion. You can see this is the developing spinal ganglion. So one developing spinal ganglion. I will write in the chat here. And what's special here? Number two, you see there's big cells inside. So large cells, if you zoom very, very high magnification, you will see cells with the nucleus and nucleolus, so large sized cells, approximately here. And these cells are called gangliocytes, big cells in the ganglion. So gangliocytes, these are the big cells inside. You also might spot small cells, so very small cells, so gangliocytes, that's two, sorry, here. And there's very small cells, look like small dark dots. These small cells are developing glial cells, very small cells inside the ganglion. So the big cells, gangliocytes, small cells, glial cell development. So only two cell types. And finally, nerve fiber bundles. Nerve fiber bundles can appear in slides. Nerve fiber bundles. So nerve fiber bundles, as you know, spinal ganglions also have nerve fiber bundles through them and around them. So you also see nerve fiber bundles. And that's the main information. So whole ganglion, of course, big cells are gangliocytes, small cells, glial cell development, and nerve fiber bundles. So glial cell development, that would be number 3, glial cell development. There's a mistake, typo, so the glial cell development, that's number 3, or developing glial cells, that's the same thing. Is the spinal ganglion understandable? Yeah? Very briefly, some other variations. Now, I will zoom out in this picture. Besides the spinal ganglion, this slide is unique. You have the spinal cord. So SC, this is the spinal cord. It has all the zones. There's even central canal. You can check the zones on by yourself. You also have vertebral column development. And you have here ear cartilaginous capsule. You can actually see the cartilage, hanging cartilage, where the inner ear and middle ear will develop. So you see the ear development partially here. This is a weird slide, but this is also a variation. So spinal cord development, vertebral column, as well as ear cartilaginous capsule appears partially here. Okay. Then what else? I will show you the microscope very briefly. In the microscope, spinal ganglions look like this. There are slides where you have separate spinal ganglions. And if you would zoom in, like I will do it here now, then we can start to ask about the cells. I will focus here a bit. So when you zoom in here, you see the whole spinal ganglion, but these cells, these are gangliocytes. Gangliocytes, small cells, glial cell development. And you also see nerve fiber bundles. So gangliocytes are the big cells. So gangliocytes, big cells, glial cell, GCDV, so glial cell development, and nerve fiber bundles. So gangliocytes, these are gangliocytes. I will write the names. So glial cell development and nerve fiber bundles. So just a slight variation. We have 5 minutes left. We need to check one of the more complicated slides, brain development. Brain development, 11, 12 weeks. Brain development. We will need to finish with that slide. Some slide variations, we can discuss them during consultation times because there's quite a lot to check. So brain development. I will put pictures side by side. This picture, yes, this picture goes here. I will decrease this in size and I will take brain development. So brain development slide. Here it is. So what is the main problem with these slides? Size, so very large size. But we will go through these slides in detail. So first of all, I will use the picture here. Let's move here. So the slide needs to be checked in zones. So first zone here, number 1, eye development. So that's number 1, eye development. So I will write, don't worry, brain development, week 11, 12. And first structure, eye development. We will talk about eye development during next class. We won't talk about each single structure there. That's for another week. But you see the eye. So the eye is developing here. So eye development. This slide is quite big, but you need to focus through the zones. We will move to the second zone here, number 2. This is nucleus caudatus. Nucleus caudatus. Number 2, nucleus caudatus. If we move in the microscope, nucleus caudatus will be visible here. So nucleus caudatus, it stands out quite well. So there's nucleus caudatus, that's number 2. Below nucleus caudatus, you see a very light area. This light area, number 3, is called capsula interna. So number 3, capsula interna. So that's the light area below nucleus caudatus. Capsula interna. Inside, you see dark strands of tissue. So you see very thin fragments here that move through the capsula interna, migrating neuroblasts. Number 4, migrating neuroblasts. Migrating neuroblasts. Yeah, they are moving through capsula interna. These dark strands, these are migrating neuroblasts. Then let's move higher, above nucleus caudatus. So if we move above nucleus caudatus, you see there's a big cavity. This is the lateral ventricle. So number 4, lateral ventricle. And inside the ventricle, you see this tissue, plexus choroideus. So 5 is lateral ventricle, 6 plexus choroideus. Then, so 5 is lateral ventricle, 6 plexus choroideus. You also see some nervous tissue here. This with number 7 would be neocortex. Neocortex. So the brain hemispheres will form. Neocortex. Neocortex. Then number 8. If you go a bit further, you will see a very thin wall with bone trabecules. So bone trabecules start to appear here. This with number 8 is called desmocranium. Number 8, desmocranium. So intramembranous ossification is happening, so new bone tissue grows, desmocranium. Then let's move to another area. Let's move here, and we'll put number 9. Here was the nucleus caudatus. Let's move to this area, just behind nucleus caudatus. This is thalamus-hypothalamus region. So number 9, thalamus, hypothalamus region. So the thalamus and hypothalamus region is a very big area here. Thalamus-hypothalamus region. So quite a big area, as you can see. Let's go. So thalamus-hypothalamus region. I hope you got this name. If we move slowly below thalamus-hypothalamus region, you will see this area that looks like a bent knee. So it looks like a bent knee here, number 10. This area is very important to remember, pons. So developing pons. This area, this specific area with number 10, pons. So pons is developing here. And there's multiple locations you need to label, 11 and 12. 11 would be, pons was here, 11 is this area. We call this lamina quadrigemina. So lamina quadrigemina. So lamina quadrigemina is here. Lamina quadrigemina. It's the midbrain technically, but we use a special name, lamina quadrigemina here. But 12, number 12 is medulla oblongata. It's here. This other part going downwards, it's medulla oblongata. Yes? Only the surface, yes. Yes, also, yeah. Only the surface, yeah. That's 11. So 12, medulla oblongata. Then you see there's a cavity, number 13. You also see the cavity here in the microscope, fourth ventricle. Fourth ventricle. So number 13, fourth ventricle, ventriculus quartus. So fourth ventricle. So ventricle number 4. And inside this ventricle, if I move down, you will also see plexus choroideus, same number 6. Plexus choroideus reappears again. Plexus choroideus. So plexus choroideus is here. Then number 14. It would be here, this fragment. Cerebellum. So 14, developing cerebellum. You can add developing to every name, but cerebellum. It also can be higher up. Cerebellum also can develop here, as you'll see in some slides, but developing cerebellum. 2 more structures left, or 3 to be exact. Here you see there's a plate of cartilage. This plate with number 15 is visible also on the microscope. So you see there's a plate of hyaline cartilage, chondrocranium. So number 15, chondrocranium. Chondrocranium, or chondrocranium. Then number 16. So 15, chondrocranium. Let's go back close to the pons, and you see hyaline cartilage in front of the pons. Ear cartilaginous capsule. Ear cartilaginous capsule. Or inner ear development. So ear cartilaginous capsule. So this hyaline cartilage here, the inner ear is developing. That's 16. And final structure, 17. This is the trigeminal ganglion here on top. Trigeminal ganglion. You see, this is the ganglion that comes from the fifth cranial nerve. Trigeminal ganglion. So this is visible here. Trigeminal ganglion. There's some additional variations between the slides. We can talk about them during consultation times before the colloquium. But colleagues, thank you very much. That's everything. Sorry for keeping you later than the class time, but these slides are quite big and I want you to be prepared for the colloquium. Okay, thank you, colleagues. You can finish. Goodbye. Okay, colleagues, those who are in the chat, thank you very much. See you later next time. Thank you very much. If something was missing, we can discuss that during consultation time. So thank you and goodbye. Yeah, sorry for keeping you that long here. Thank you.